Okay, hello and welcome everybody. My name is Gloria Isabel Giraldo Calderon, and I will be one of your instructors today. Before we get started, let me introduce you to Dr. Marianne Mcdowell from the University of Notre Dame, who is one of the 3 co-PIs of the ViewPath project. Thanks for being with us today, Marianne.
Thank you so much, Gloria, and welcome everyone. And thank you for joining us this morning for the second in a 4-part webinar series. As Gloria mentioned, I'm one of the joint PIs of the ViewPath DB project. And many of you may already know that I took over the PI position from Frank Collins in the previous iteration of the VectorBase resource. So this might be a repeat for some of you, but first I'd like to remind you that VectorBase has always been part of the Bioinformatics Resource Center program, or the BRC program, funded by the National Institutes of Health. The program started in 2004, and there were originally 8 BRCs, and the funding cycles go in 5-year increments. And in this last contract cycle, the NIH only offered 2 contracts, one for a BRC for bacteria and viruses, and one for parasites and vectors. And so we merged with UPathDB to become ViewPathDB. We started merging in this process in September of 2019, and we are just about complete, which means that the legacy VectorBase that we are still currently maintaining will be taken down in May of this year. So if there's anything you want to do on that, you probably better do it quickly. Even though we were forced to merge by the NIH, we're quite excited because this combined resource leverages the strengths of both VectorBase and UPathDB. And the topic of today's webinar is one of those strengths from UPathDB that we now have made available for vector users. And so I think you'll find the search strategies really helpful for your research. So I know that change can be difficult, and we appreciate your patience, and I'm glad that you're here for this training today. One of the things that also was an advantage to vectors is we merged our outreach teams. So we still have Gloria, but now we have the outreach teams from UPath as well. It's an excellent team, and I'm sure they'll be able to answer all of your questions. So thanks again, and I hope you enjoyed today.
Thank you very much, Marianne. Now, some logistics. This webinar will last for 1 hour. This is the outline for today. And the main goal is to answer your questions. Our main topic is how to construct a search strategy, as Marianne just said. And we will do it with you step by step using different examples. Not only during this webinar, always the outreach team is ready to answer your questions and hear your feedback using all these communication channels. The email in the link for contact us, the community chat at the bottom of the page, and our social media pages, Facebook and Twitter. We are recording this webinar, and you can access to our YouTube channel and select in the playlist the ViewPath webinars playlist to see the recording.
Hey, Gloria, Gloria, sorry, I'm only seeing the first slide from your presentation. I'm not sure if that's only me.
Stop sharing. Sharing again. Do you see the slide now?
Yes, that's better.
Okay, thank you, Omar, for letting me know. So I just said, this is how the panel looks to you, and all the questions you type here are private, and Omar and Susan in the background are going to answer the questions for you. And if you're on the homepage, not only during the webinar, but anytime, we welcome your questions. You can contact us, social media, Twitter, and Facebook, and here at the bottom of the page in the community chat. And here is the link to the YouTube channel. You arrive and select webinars, and that's where all our recordings are. And also, you can do it this route. Click on Help, learn how to use ViewPath, select the webinars, and we have the list of the upcoming webinars. These are the ones from this series, the ones for VectorBase, but also is the list from the previous webinars, and there you will find all the recordings. So as described in the invitation we sent out, this is an introductory webinar series, and we will use vector-based examples only. But because the layout of all the ViewPath sites is the same, you can do anything that we are going to demo in any of the other ViewPath sites or parasites or fungi, using different data sets, but the same tools and layout. So if you look at the bottom of all these of the sites, you will find a link to move to all of them. So for example, here I'm showing you Pitoplasma database. And here, this one here is the one for VectorBase. The icons to the right, their layout is a little bit different, but they are also part of the View project. So OrthoMCL, you can use it to investigate gene homolog relationships of all the genes in the parasite, fungi, and vector-based websites. The other one is CleanEpi database for the exploration and analysis of epidemiological studies. And the last one is microbiome database, which is a data mining platform for interrogating microbiome experiments. Okay, so today the instructors are Omar Harp from the University of Pennsylvania, Susan Warrenfelds from the University of Georgia, and I, Gloria Isabel, from the University of Notre Dame. Each one of us will demonstrate how to construct strategies with different searches, which will cover these major points: starting your first step, adding more steps, Boolean operations, transforming results to orthologs, and downloading and exploring those search results. We have four very important take-home messages from today. One, the strategies we are going to construct step by step with you today are just a guide that for the types of strategies or number of steps you can add, and that depends on your research question and your creativity. Second, you can use the obtained results for many different purposes as part of an exploratory or pilot project, upon which you can continue with wet lab experiments. You can use the strategy results to characterize a list of genes you have found in your vivo or in vitro experiments, or you can base your complete research just using bioinformatic resources from our website or other websites. Third take-home message, you can construct a strategy using in silico predicted data, and we are going to demo that, like transmembrane domains, signal peptides, protein domains, but you can also use in vivo or in vitro generated data, such as microarrays and RNA-seq. And the last and fourth take-home message for today, if the data you want is not on our website, please contact us, and we will discuss feasibility of inclusion and timeline. The data can be yours or from other scientists that have been already published. So now I'm going to show VectorBase, and Omar and the others, please let me know, are you seeing now my VectorBase screen?
Looks good.
Thank you. So in the previous webinar, I presented to you the very basics of the searches and how to use them to construct a strategy. That time I constructed a two-step strategy. Today, I will add one more step to that very same one. If you did not attend last time or do not remember what we did, no worries. I will go very quickly over the first two steps. This time, so that day, I used this menu here on the side, but you can also use this one here on top. So as keyword, I'm going to use gene ID and select that one from the list. That day, I copy from my list of genes here in this box. Today, I want to upload it. So I'm going to select, this is my text file, how it looks, and get answer. And here I have my list of genes, and I want to add another step. Click there. And what I'm going to do is I'm going to transform these genes to orthologs, and I can select any from all the 40 or for the 45 genomes available in VectorBase. I'm going to select edits, for example, and I'm going to do run step. And for the third step, I'm going to add RNA Seek Evidence. And this is a study that that I presented to you last time. the neurotranscriptome of Aedes aegypti, different tissues, male and female, and I select fold change. And here, notice we have a lot of options, so you can always increase the size of your window. I am going to select, my references are going to be males. 1, 2, 3, 4, 5, 6, 7. All the 7 male tissues. And for the females, I'm going to select the non-blood fed. So I have here 2 non-blood fed, 2, 3, 4, 5, 6, 7, 8, 9. I'm missing 1. Oh, well, okay, I'm gonna run it like that. Okay, so here we have it. This is a vector-based strategy, and it has these 3 steps. And now you can save it, and you can click here and provide a name. I'm just gonna say test and saved. And you don't have to be logged in to use VectorBase, but notice I am. And this allows you to save and to share your strategies. And by default, all your strategies are private, and you have to select if you want to make them public or not. But if you save them, this is another way to keep your work records, in addition to the personal notes that you have in your lab notebook. Previously, I have also demoed all these actions here. I'm not going to show you that today. I'm skipping that. And now I'm going to show you how to explore the data. So I'm going to click on the first step. Notice the box turns yellow, and this tells me that I have started with a list of 12 gene IDs. I hover with the mouse and notice how the edit option appears. And it says, it provides the details of what I did. And here is reminding me that I have uploaded this list of 12 genes. So I can close this. Now I want to explore these results, and I'm going to click here in hide organisms because I don't need them. And by default, you're always going to have gene ID and transcript ID. But you can add more columns, and what I have done previously is I click add columns, and I have selected gene name or symbol or product description. And VectorBase is remembering my preferences, and that's why it's showing me that. I now want to click on the second step, and notice now this one turns yellow. And again, if I hover with the mouse, I click on edit, and it's telling me that I generated the orthologs to Aedes aegypti. I close this here, and now I'm exploring the results. Notice this table change when I select the first or the second step. And now, because I transformed to orthologs, I added a column that is called input orthologs, because that's the one that matters for me, looking at the second step. And remember, for the orthologs, you have these 45 organism genomes in VectorBase to choose from, and please select the ones that are biologically significant for your research question, remembering that their evolutionary distance matters. Okay, and now on step number 3. So here it says that I have 2,989 genes. So I click on edit, and it's just telling me exactly what I did, the title of my experiment. Oh, I did not change this, but I can say, okay, I just want the genes that are upregulated, for example, and I can revise that. Okay, and now notice my number of genes have changed. So it is just saying that for the parameters that I selected, the tissues that I selected, I have available these ones, but those are not all the ones for that experiment. It's just for what I chose. And this is the genome of Aedes aegypti is about 14,000 genes, and you can find that out if you go to data, organism, genome information and statistics, and there you select Aedes aegypti. And when you are on the page, it will tell you how many genes this one has. And in my strategy, what I said is I am working with the protein coding genes only. I could also revise that and change it, but that's what I'm selecting to work with. Now I'm going back to the second step to show you how you can download this data. So you have different options. Notice that here I have selected input orthologs. So if I click on download, I come here, and that option that I have already selected, input orthologs, is chosen here by default. And I can select if I want to download this data as, for example, a CSV file, which I can open in Excel, but right now I'm just going to select show in browser. And here I have my file. I can also say, no, I want the FASTA sequence in protein format for these genes. And again, you can have a text file, but I just want to see it in browser. And here you have it. Okay, so now that we have run through this first strategy, I'm going to switch gears a little bit. And with this strategy that we just constructed, I hope that I have given you a clear idea of what a strategy is. And now I'm going to give you a little background in an effort to construct your own strategy. So here again, I'm in the home page. And as I said, these here from here, the searches are exactly the same ones. So these are the many different ways in which you can start a search. And these fall in these categories: genes, organisms, genomic sequences, genomic segments, ESTs, metabolic pathways, and compounds. What these categories mean is that they are the types of results you are going to obtain. If you select this one, like I did when I did gene ID, so I obtain a list of genes. If I start from the organism one, I'm going to obtain a list of organisms and so forth. So if you go to the public strategies here, you click here, my strategies, and click on the public ones. We have created 20 for you to use, and the type of results that they return, you found them here in this column. So we have ones that returns data sets, returns genes, metabolic pathways, and organisms. So these are just some examples to get you started. And the one that I just demonstrated is this one here, demo number 1. Okay, go back to the home page. If I expand them all, you can see all the different options that we have, but you don't have to memorize them. It's just, I suggest you to take a look so you know what is available to create your own steps. Just, it's like having a table of contents of a book. If you go through this, you know what to expect and how you can start your strategies. So for example, if I type a keyword here, I'm going to type text and select this strategy here. And I'm going to say, I want to do one with Anopheles gambiae. Oh, I'm going to open this. And I'm going to do Aedes. So notice this has changed. Now I have 2 selected. I want to do bullets. Here it changed. Now I have 3. And you can change your name. And notice this has a wildcard. Why? Because you can type, for example, the word kinase, or you can type it with a wildcard at the end, or you can type it with the wildcard both at the end and the beginning, or just at the beginning. And you can have all these fields here open, but just in the interest of time, I'm just going to select gene name or symbol, and I click on get answer. And notice this is the strategy we just ran. I'm going to revise this. Where is the product descriptions? Here we are. Sorry. So you can revise your strategy and go back. And you can save this for your future reference of how you construct it. Now I'm going to do something different. So I'm not going to use data generated in the lab, but now I want to construct another strategy, but using in silico data. I want to start one using Interpro. So I select here Interpro. And here in Interpro, this is a database to classify proteins, especially useful if you want to describe a list of genes and maybe not all have gene metadata, I mean gene name and symbol and product description. So that's very useful for you. And here I'm going to select Anopheles. Select this one. Notice I have selected one. I want to select from Interpro and which type of genes I'm interested in, GPCRs. I select one of the categories here and I click on Get Answer. Okay, I have obtained 82 genes. And now I want to add a step. And these categories that we have here to combine with other genes are exactly the ones that I just showed you in the home page that are for the gene categories. And what I'm going to do is I'm going to combine this with transmembrane domains. So I have here transmembrane domain count. Again, I'm working with Anopheles gambia pest. I select that. And because I want to work with GPCRs, I know those genes, those proteins have 7 transmembrane domains. So I only want 7 and I run a step. But I have done an intersect and that's not what I want because I don't want to have what is common between the two. I want to have both of them. So I edit this and I said I want the union of those two types of data. So I revised my strategy and notice how this number here has changed. And for step number 3, I can say, for example, I have a list of gene IDs that I want to know if, do I have GPCRs in my list of genes? So I choose a file and here I download this data from a paper and I run the step. And here I have the genes that I have uploaded. How can you get those lists of genes? For example, I went to this paper and I just copy all this list of genes here. And here you have it. So from the genes in VectorBase that have interpro domains consistent with GPCRs, and those genes with 7 transmembrane domains, I look in my list of genes, which ones of my genes seem to be GPCRs, and here I got the list. Okay, so now I'm going to switch over to Suzanne.
Okay, can you see my screen?
Yes.
Yes, we can see.
Okay, actually that's not the screen I want to show, I think. Okay, so if you all look in your handouts panel, you'll find a document that will take you step by step through what I'm about to do. And it offers a little bit more explanation of the concepts. So if you want to download that and do it a little bit later, it probably would take you about 45 minutes, but it's a good exercise to learn overall what the strategy system can do. Right, so today I'm going to pretend that I'm a researcher who's interested in the regulation of mosquito midgut genes that have protease activity. And my organism is Anopheles gambii, so I'm going to try to produce a list of Anopheles gambii genes that are proteases, that are expressed in the midgut and have transcription factor binding sites in their upstream regions. We can build a strategy to find these genes that have these 3 biological properties. The strategy that I'm going to build will look like this. So each of these searches queries a different data set that has data that speaks to a certain biological property. We'll have 2 searches that return proteases, and we'll add those 2 together. Then we'll query some RNA seek data and pull out those proteases that are expressed in the midgut. We'll transform those genes into Anopheles gambii, because I'm a gambii researcher, and then pull out those genes that have DNA motifs in their upstream regions. And in the end, we'll have Anopheles gambii genes that are proteases expressed in the midgut, and also have DNA motifs that represent transcription factor binding sites in their upstream regions. We'll end up with about 18 genes. So let's go to VectorBase and start that. We'll start with a text search, right? The data that we're searching here is gene annotations. We'll go search for genes based on text. And what I find is that I have several parameters that I need to fill in before I can click get answer and run a search. So what we want to find genes that might be proteases. So we'll do a text search and look for genes that have the word protease associated with them somehow. I'm going to go ahead and search all of the organisms in VectorBase. My text term will be protease. And then the fields define where in the gene record to look for the term protease. And I'm going to leave them all clicked. Click get answer. So it could be that the, sorry. Okay, I'm sorry, I'm having a little technical difficulty. So we have run a text search for the term protease in all of the organisms across VectorBase. And the search returned 20,181 genes. Now, I can look at the distribution of those hits across all of the organisms here. And it looks like we have between, I don't know, 200 and 700 genes per organism that have the term protease associated with them. And then this is our list of 20,000 genes. It's a good idea after you run a search to sort of look at what you get back and try to say if you think the search worked, because if it didn't, you can go back like Gloria showed you and change some of the parameters. I like to look at the product description. I see some that some genes that are not defined well enough to have a product description. We don't really know what it does, but then here I also see a bunch of genes whose product description contain the word protease. I'm not really worried about these unspecified genes. It just means that the search found the term protease in one of those other many fields that we searched. So I'm going to call this a success, and I'm going to look for more proteases. There is other data in our site that indicates that a gene protein product might have protease activity, and one of those is the gene ontology annotations. They are a controlled vocabulary of terms that curators can actually tag onto a gene. There are 3 ontologies, one for molecular function, one for biologic process, and cellular location. So we've loaded all of the Go annotations for all of our organisms and created a search that returns genes based on their Go assignments. So let's add that search. We'll click Add Step. I want to combine with other genes, and then we have 2 other things that we need to do, right? Choose how to combine the genes and choose from what search we want to use. Now we notice we have several choices about how to combine our text search results with our Go term search results that we're about to run. Let me talk about that a little bit here. So we'll end up with 2 lists of genes, right? A text term result list and a Go term result list. And just in mathematical terms, there are only a few things that we can do with these 2 lists. If we intersect them and we will find genes that are in common between them. We can union them to say, just give me all of the genes in my next result. And we can subtract them. We can say, remove all of the genes that appear in this list from this list, so that I only have genes that are unique to list 1. We could do the opposite. And then we can also, because we know the genomic location of these features and we know the location of these genes, right? We can say, show me genes in this list that are close to these genes in that list. And there's some special circumstances where you'll want to use this, and we'll get into that later. But right now, what we're doing is we're trying to create a big list of proteases. So we want to union our new search with our old one. So I'm going to choose to union from our GoTerm search. And I'm taken to a pop-up that has the parameters I need to fill in before I can query. So I'm going to select all organisms, use default on these. And I know that there's a Go term that is defined as proteolysis. I guess I need to scroll down. There we go. Proteolysis. And there are many other Go terms. If you're interested in that data, you should learn a little bit more about the ontologies and what Go terms really define the properties of the genes that you're looking for. But so now I have it all set up. I have my organisms, my Go term, and I'm going to click Run Step. So I see this, my Go term search returns 27,000 genes, and the union of my Go term and my text term is about 35,000 genes, right? So now I have genes that have two biological, well, I've used two different lines of evidence to produce this list of protease genes. Now, the next biological property that I want my genes to have is I want them to be expressed in the midgut. So we can search our RNA seek data to find what we need. So I'll click Add Step, combine with other genes. And here we are again at that need to combine, we need to tell the search how it's going to combine our new list with our old list. And let's think about that for a moment. Here we go down. And just to expand on what we said before, so we have a list of proteases, right? And we're going to create a list of midgut genes. And we can see here by these little dots that there's only 3 genes that are in common in both lists. Right, so if we intersect that, we will end up with only those 3 genes. And these genes will be proteases that are expressed in the midgut. So let's go ahead and do that. We're going to intersect that with some RNA seek data. And the first thing it's asking us to do is to choose our experiment. And it turns out that we have this perfect experiment that was performed by Tomas et al. In 2016 that looks only at the midgut transcriptome. So I'm going to choose that search. And I'll configure that search to show me midgut genes that are highly expressed in that sample. So between 90 and 100 percentile. And I'll click Run Step. So this is going to produce a list of genes. It produced about 1,400, almost 1,500 genes that are highly expressed in that midgut sample. And the intersection with our protease is 108 genes. So these 108 genes are proteases expressed in the midgut. Now, if we look at the distribution of those genes, we see that they are only expressed in Anopheles crucifices. I'm sorry if I'm pronouncing this wrong. So there's, we have so many zeros here. All of these organisms did not return any genes. And that's because this RNA seek data was performed in that one organism, Polycypheses, and so the data was aligned to that genome, and cannot any IDs in this list there are no like Gambii genes in this list that can be combined with this. But what we can use the orthology transform tool to turn these back into Anopheles Gambii genes. So I'll say add step. I want to transform based on orthology into Gambii. And click run step. And so these 108 genes turned into 217 Gambii genes that are orthologs of these. Now we're using a little bit of, we're inferring, right, these biological properties onto these Gambii genes. And these would be proteases expressed in the midgut. Now, the last thing that we wanted to do was to look for transcription factor binding sites in the upstream regions of these genes. So we have a, we're interested in the regulation here. We have a DNA motif search for finding motifs anywhere in the genome. The thing is that this search will not return genes. It returns segments, right? So let's think about that for a little bit. Here we have a list of IDs from genes and a list of IDs that are segments. And they're apples and oranges, right? There are no segment IDs that are in this list of gene IDs, right? It just doesn't mix. So there are no IDs in common. You can't use any of these Boolean operations. But what we do know about each of these is the genomic location. We know where these are on the genome, and we can use our collocation tool to say, show me genes from this list that have these little segments in their upstream regions. So let's do that. We are going to use the genomic collocation tool on a new search for a DNA motif pattern. I'm going to do that in Gambii. And it turns out that this is a transcription factor binding site, the default pattern that's in here. If you guys have motifs that you want to look for, you're going to need to express that as a regular expression and then put that in here. But for right now, I'm going to use this default transcription factor binding site here and say continue. And then I need to set up the collocation, right? And there's a little sentence here that you need to fill out. Return each gene whose upstream region contains the exact region of my genomic segment, which is my DNA motif, and is on either strand. So I can click run step. Now, what it's doing is searching the entire genome of Anopheles Gambii pest for that 20 base pair segment, and it turns out that it returned 27,600 segments. And there are 18 genes that have this segment in their upstream regions, right? So we have now 18 genes that we can take to the lab and do our experiments on. And those genes we think are proteases expressed in the midgut that have a certain transcription factor binding site in their upstream regions. And I remind you that there's a handout that you can download that takes you step by step through this.
Thank you, Suzanne.
Yep.
I'm transferring now to Omar.
Okay, great. Thank you. So hopefully you can see my screen. So again, I'm showing VectorBase. This is the same strategy that Suzanne just walked you through step by step. And the feature I'm going to show you today, actually, I'm going to show you a couple of features. One is, let's say, Suzanne saved this strategy. It's actually shared in the public strategies as well. So any one of you can go to the public strategies right here and select it and open it up in your own workspace and play around with it. But Suzanne could also share the strategy by saving it and clicking on this share link and emailing the link to me, right? And so let's say she sent it to me and I open it up and I decide, oh, you know what? I'm going to modify this a little bit. I have some other ideas, but I don't want to mess with this strategy first because I want to keep the original as is. So the first thing I'm going to do is I'm going to duplicate this strategy. And so right here in the strategy panel, there's this icon, the copy icon. So I'm going to click on this, and it tells me, do you really want to make a copy? And I say yes, make a copy. And so what will happen, it's going to duplicate this strategy exactly as it is, and you'll see a second panel as this happens, showing you the same exact strategy, which now you can modify without messing around with the previous one. So you'll see here it has the same name, proteases expressed in the midgut with upstream motif, and it says a copy of that. You can modify this name if you want, or do whatever. So now I was looking as Suzanne was working on this strategy, or when she sent it to me, I thought, it would be cool if I can add additional types of data, additional experiments to this midgut transcriptome. Because as you remember, if we click on edit here on this particular step, you'll see that she's picked a specific RNA seek experiment from a particular species of Anopheles. And I know there's a lot of RNA seek and microarray data in the database. So I'd like to combine more of those data sets together because I want to, leverage the power of multiple experiments. Hopefully you've seen over the demo that Gloria and Suzanne gave you that the strategy system really gives you this power to combine a variety of different data types and put them together and hopefully come up with some meaningful results that you can take back to the lab, develop some new hypotheses and work on them. So what I'm going to do is, if you imagine this strategy as a branch on a tree, I'm going to take this little leaf on this branch, I'm going to expand it some more. So I'm going to add a few more steps in this leaf and then feed back into this strategy because I want to add more experiments up here that I'm interested in. So I'm going to click on edit. And once you click on edit, you'll see here there are a number of options that you can pick from. One of them is called make nested strategy. So what happens when you click on this? So as soon as you click on this, it takes this step. You'll notice here there are 1,473 genes in this result. The same ones are shown up here. So it's basically a zoom into this step right here. And now I'm going to add additional steps and grow this nested strategy, the results of which are going to be fed back into my main strategy. Okay, so let's go ahead and add a step right here, just as Suzanne and Gloria showed you. And I'm going to look for microarray experiments. And I just typing in the search of searches, basically. I'm searching for microarray and immediately it filters the results. I'm going to click on microarray evidence. And as you've seen before, I'm going to click on midgut and you'll see a bunch of experiments show up. Again, it doesn't matter which one you pick. You obviously, hopefully, when you do this, you'll look at the paper a little bit and decide this is the experiment I want to choose for my work. So I'm going to pick this last one right here. Again, I'm going to use the same method that Suzanne did. I'm going to pick percentile. One thing to note is I didn't do in the beginning, but you can always do this. It's set to intersect. But in my case, I really want to use union. I want to basically get all the genes from all the experiments that meet the criteria that I select. So I'm going to change this to union. And as we scroll down here, I'm going to select the midgut. This is a microarray experiment. I could look at this and say, I know Suzanne picked them above the 90th percentile, but I may say, well, for this experiment, based on the data and based on the paper, I'm really going to change this to a 70th percentile because I think anything above 70 is really highly expressed for this particular experiment. And that's obviously something you would have to do and look at. And I'm going to click on run this step. So what's going to happen is going to find all the genes that are expressed above the 70th percentile in the midgut stages from this experiment, and it's going to union the results, so 707 genes. It's going to union them with my previous midgut transcriptome, which is an RNA sequence experiment, right? So I'm combining two different types of transcriptomics experiments, The result now it's 2,180 genes, and now you're intersecting these results with the results from the Go term and text search. And notice that the results downstream update as well. So you don't have to replicate everything. Now, instead of 18 genes, I have 20 genes. And you can grow this as much as you want. So I can go back here. I'm going to pick union right from the start. I can pick RNA Seek now. And let's pick RNA Seek. And again, just as Suzanne did, I'm going to type midgut. And again, there are multiple experiments here. And instead of picking this species right here, I'm going to find one in Anopheles gambiae. And let's pick, doesn't really matter which one we pick, but let's go ahead and pick this midgut one from Gomez-Diaz 2014. I'm going to pick the percentile. And again, clearly you want to go in and learn more about the experiment. So I haven't specifically looked at this particular experiment, but I'm going to go ahead and pick one of the samples. And I'm going to say I would like it in the 80th to 100th percentile. And again, you run this step. The nice thing about this is that this is an in silico experiment, right? We're doing this in silico experiment. And if the results don't give you anything interesting or give you 0 results, no big deal. You haven't wasted any reagents. You're just manipulating the web browser. It's a little bit of your time. And the nice thing about it is that you can go back and you can modify any of these steps, delete them, revise them, and so forth as you go on. So now you'll notice my gene number increased. Every time you use a union, your numbers will increase. Every time you use an intersect, your numbers will decrease. That's basically how it works. But in my case, of course, my numbers increased. And now I'm up to 22 genes that meet the criteria that I'm interested in. Okay, so I think I will stop here, Gloria, and turn it back to you.
Okay, Omar, thank you very much. So I think we did really good on time today. So as we all worked you through today, so I started with really, simple way of constructing a search. Suzanne took it a little bit further and show you a little bit more complicated strategy, and Omar just went through for the nested one, which we think is the most complicated. So thank you very much, everybody, for attending today's webinar. If you did not have time to type your questions or you have not got an answer, please contact us via email or the community chat or the VectorBase page and send us a message. And see you in about a month for a 3rd event of the series, in which we will go even deeper into the transcriptomics and how to analyze that in the genome browser, in the gene pages, and more in depth when you are looking at the genes that are upregulated or downregulated, how to actually extract the data from VectorBase. Thank you very much, everybody. Bye-bye.
Bye.